For most of medical history, the presence of bacteria in the bloodstream was a flashing red alarm, a sign of infection, invasion, and impending immune warfare. Blood was assumed to be sterile, a pristine river that only turned dangerous when infiltrated by microbial outlaws. But now, as science gets better at detecting the invisible, a more nuanced picture is emerging, one that's less invasion of the body snatchers and more like a shadowy diplomatic dance between host and microbe. In fact, under certain conditions, bacteria, or at least their byproducts, may not only coexist in blood but also participate in tuning immunity, vascular integrity, and even longevity. That's right, the bacteria in your blood might be trying to kill you, or they might just be helping you age more gracefully. Let's start with the usual suspects: pathogenic bacteria. When microbes like Staphylococcus aureus, E. coli, or Pseudomonas aeruginosa enter the bloodstream, it triggers an immune firestorm. These infections, often originating from the gut, lungs, skin, or surgical sites, can lead to sepsis, a systemic overreaction where the body's inflammatory defenses spin out of control. Cytokines like IL-6, TNF-alpha, and interleukin-1 set surge into the bloodstream, causing blood vessels to dilate and leak, organs to lose oxygen, and tissues to fall into crisis. Even if the infection is cleared, this intense immune activity can leave behind scars, metabolically, cognitively, and immunologically. Repeated low-grade infections or chronic microbial translocation, especially from a leaky gut, can drive what researchers call inflammaging, a smoldering state of systemic inflammation that accelerates vascular aging, weakens metabolic flexibility, and dulls immune precision. But not all bacteria in the bloodstream trigger alarms. Recent studies using ultrasensitive DNA sequencing have found traces of bacterial DNA and sometimes intact bacteria in the blood of healthy individuals. While skeptics rightly warn about contamination, the consistency of findings suggests there may be a resident blood microbiome, or at least transient microbial passengers that modulate immune tone without triggering full-blown immune response. The line between harmful and helpful may not be based on presence alone, but on context, balance, and bacterial behavior. Take, for instance, the newly studied Paracoccus sanguinis, a blood-resident bacterium with intriguing, possibly immunomodulatory properties. Unlike pathogens that provoke inflammation, this species may help regulate immune tolerance, much like gut microbes train the immune system not to overreact to harmless food particles. If future studies confirm that certain bloodborne microbes promote regulatory T-cell activity, suppress oxidative stress, or fine-tune endothelial responses, they could become allies in preventing atherosclerosis, insulin resistance, and tissue fibrosis, hallmarks of aging gone wrong. Meanwhile, bacterial metabolites, especially those derived from gut flora but circulated via the blood, are another layer of this microbial mystery. Compounds like indole, 3, propionic acid, butyrate, and secondary bile acids don't just passively float through circulation. They bind to receptors on immune cells, influence gene expression in vascular tissues, and even cross into the brain to modulate mood and cognition. Some, like indoles, reduce oxidative stress and DNA damage. Others, like lipopolysaccharides, LPS, can ignite the innate immune system, causing endothelial dysfunction and chronic inflammation. Whether these molecules heal or harm depends on dose, duration, and host resilience, a balancing act more delicate than any detox plan can manage. Perhaps the most fascinating future avenue is the idea of harnessing bloodborne bacteria, or their products, as therapeutics. Imagine engineered microbes that release anti-inflammatory exosomes or antioxidant peptides directly into circulation, acting as living delivery systems. Or customized postbiotics, refined from microbial metabolites, that improve mitochondrial function or stabilize vascular tone. In this vision, the bloodstream becomes not a battleground, but a distributed platform for microbe-host collaboration, where aging is not only slowed but actively modulated. Of course, we're not there yet. Current tools struggle to distinguish contamination from colonization, and immune responses vary wildly between individuals. But the concept is gaining traction, that microbes in the blood aren't always intruders. They're sometimes informants, builders, or even guardians. The next generation of anti-aging strategies may no longer just fight bacteria with antibiotics or disinfectant metaphors. Instead, We may be studying how to curate the blood microbiome the same way we cultivate soil microbes in regenerative farming, balancing ecosystems to grow resilience, not just sterilize dysfunction. In the end, the bacteria in your blood may well be writing parts of your aging script, some in the language of damage, others in the grammar of repair. Whether you get the horror story or the healing narrative may depend not just on what's present, but on how well your immune system can interpret the microbial message.